Hello and welcome with another video. Right now I'm in my office. Today is the lab day, so I'm going through this checking process of this work. So there are lots and lots of journals which I need to check to see whether they are inputting the data in the right format or not, because that's a must. Now at least they learn to draw the figures. I mean the figure of there are no such figures. It's not biology or zoology, botany, it's chemistry, but you just need to, I just told them to draw the assembly setup on their journal, so that they must remember how to set up an assembly for different reactions, or for different, I mean, type of tests. So this is what I am doing right now, going through the checking process of their journals. And there are piles. This pile I have checked, these journals I have checked. Now these are the remaining ones which I will soon check. And today again I'm going to assign them a new, I mean, experiment, and they are going to start with the experiment number 4. Up till now they have done 3 experiments. The first week was the general introduction week, then I have started, I mean, the actual work, and they were, according to their syllabus, they were assigned the determination of different melting point tests, melting point determinations of different compounds, organic compounds. So they did 2 experiments for that is melting point experimentation. And then now I jump to the next, I mean, experiment, that is the boiling point of different organic solvents. So now, last time they were given an unknown sample. Today I will tell them the name of the compound so that they can easily find out the boiling point through capillary tube method. So we can use a tale tube also, but that was not available. So I just explained them how to use how to do this procedure via capillary tube method. So hopefully they've understood. And today I will ask them to do it independently. Everyone should do it independently because it's not a big deal. Just look at this figure. They simply don't know that the level of this should be the same. So I need to draw even these small, I mean, things, because this is the assembly setup they are supposed to, and such type of journals are also here where there is nothing inside the, I mean, ignition tube. There is nothing inside the capillary. What type of figure is this? I don't follow. Anyway, I'm going through this checking process. There's still time. It's 10:15, so my lab is at about 11 o'clock. So see you in the lab now. And welcome with another video. I'm in my BS Part 2 lab, organic chemistry major, and I just explained them the 4th experiment. You can see behind my a blackboard in which I have explained my students how to use a tale tube. Even I explained them this also, how you're going to heat the arm of the tale tube. And then I explained them how to record the melting point. When the bubbles there, they will emerge, and when they will seize down the solvent that is they are analyzing, it will start arising inside the capillary tube, and so they will record the melting point at that moment. So I explained them everything. So it's a known organic compound. Today I gave them this one, that is aniline. It is also called phenylamine, and the phenyl group is attached directly to the amino group. So my students are performing now. They are now going to, because they know the name of the compound, that means they know everything about it. So they are now performing. They are now going to make the assembly setup for recording the boiling point of aniline. It's experiment number 4 today. Thank you. These are my students. Now they are working. You can see them. This is the girls' side. They are now setting up the assembly. And this is the boys' side. It's again a very warm day. Unfortunately, whenever they have a lab, it's a damn warm day. They are negotiating among themselves how to set up the assembly and everything. So that's it. The other group is there. They are also engaged in designing how to set up the assembly. The last group, you can see, they are also involved in inputting the data. This is Aniline. My girls are now, you can see, tying the thread. Thread shouldn't be that long that it should be that it would be immersed inside the paraffin oil. It has to be very short. या उसको cut कर लें. Just cut it down. Sealing the capillary with red. Why your burner is on and your assembly is not even set up, and setup is not finished, and your burner is on? Why? You need to close the burner. First, arrange your assembly setup properly. Your assembly setup is wrong, totally wrong. Your thermometer is touching the side of your beaker. This is wrong. I explained my students. Now they are performing the lab experimental session, and I'm out for drinking some water.